receptors, they are either specialized cells that detect specific types of stimuli. Like we said, the photoreceptors in the retina of the eye, or the chemoreceptors in the taste buds, in the papillae on the tongue, pressure receptors in the skin, pressure receptors in the skin are endings of sensory neurons. The Pacinian corpuscles. So the receptors are either specialized cells, or they are the endings of the sensory neurons themselves. Okay? In the Pacinian corpuscle, which is the pressure receptors in the skin, this is an ending of a sensory neuron. But the photoreceptors in the eye, or the taste receptors in the tongue papillae, these are specialized cells. Okay? Last lesson, I told you how an action potential is initiated if it's an ending of a sensory neuron, like the Pacinian corpuscle. We said that the Pacinian corpuscle is the ending of a non-myelinated sensory neuron surrounded by a capsule. So this is the capsule, and this is the non-myelinated ending of the neuron. And we said that when pressure is applied, it will result in deformation of the plasma membrane, and sodium ions will start to leak in, depolarizing the membrane. This is known as the receptor potential. And if the receptor potential reaches threshold, it stimulates the voltage-gated sodium channels to open, so there will be an influx of sodium ions to generate an action potential. Okay? In case of the taste buds, the tongue is covered by many papillae. This gives the tongue the rough surface. And these papillae are made of many taste buds. And each of these taste buds is specific to detect a different taste. So taste buds for salt is different than taste bud for sugar, it's different than taste bud for sour. Each taste bud is specific for a certain taste. Okay? For example, the salt taste buds, when we eat salt, the sodium will directly diffuse into the taste bud in the sensory zone, and it will start to depolarize. It will start to depolarize the membrane of this area, and this will result in calcium ions diffusing into the cell body. This will lead the vesicles containing the neurotransmitter to move and fuse with the plasma membrane of the cell body, releasing its contents outside. It's as if a synapse is taking place. And this generates an action potential in the membrane of the sensory neuron, which is next to it. Like you see, this is the specialized cell, this is the taste bud, and very close to the taste bud, we have the ending of the sensory neuron. So there is a sort of a mini synapse between the sensory, the specialized receptor cell, and the sensory neuron.
Is this mini synapse?
It's a synapse. It's as if a synapse. It's exactly like the synapse. But in here, it's directly the sodium in the table salt. It diffuses directly, depolarizing the membrane. But it opens the voltage-gated calcium channels, and they diffuse in stimulating the vesicles to move and cause exocytosis of the neurotransmitter, which will initiate an action potential in the sensory neuron. the same way as it happens in the synapse.
شو ما بعد ما الصوتين بيدخل ايه calcium ions؟
It's like exactly what happens in the presynaptic neuron. مش انت في presynaptic neuron, when the depolarization reaches the presynaptic membrane, it stimulates the voltage-gated calcium channels to open, with calcium ions diffuse into the knob, and stimulates the vesicles with the acetylcholine to move and fuse and cause exocytosis. But it's the same. When the sodium ions diffuse in, it stimulates the calcium ions to diffuse in, opening of voltage-gated channels for calcium ions, so they diffuse in. And this will result in the exocytosis of this neurotransmitter, generating an action potential in the sensory neuron. Okay? Sugar receptors, they are different. If we are eating sugar, sugar will act in a different way. The sugar has specific protein receptors on the plasma membrane of its taste buds. And when it binds to these specific protein receptors, it will cause cell signaling. It activates the G protein, which activates the enzyme that forms the cyclic AMP. It will be like a process of cell signaling, which will end at the end in the exocytosis of the neurotransmitter and formation of action potential in the sensory neuron. Okay? That's the difference between them. يعني الصوديوم اللي في السول directly diffuse into the membrane depolarizing it. لكن الشوجر بيشتغل بالسيل signaling.
يعني هو نفسه signaling molecule.
هو نفسه signaling molecule. ما بيخش جوه السيل. هو بيمسك في البروتين ريسبتورز وبيستيميوليت الجي بروتين والسايكليك AMP. في الآخر الانزيم cascade leads to the release of the neurotransmitter.